Hey guys, waiting for the stream to go live. Alright, I guess we're live. So we can begin with this session. Just give me 1 second. So today's session it's on lipid metabolism, and this is part two. Alright, this is lipid metabolism part two, and the topic that we are covering today, is lipid transport, the various apolipoproteins and dyslipidemias. Right. So in this session, we are going to talk about some of the most important portion in lipid metabolism, right? So, see, try to understand that what we had discussed in the previous session, that was about a general idea of all the lipids, the various metabolic processes, including ketone bodies, the synthesis of ketone bodies, and everything around that. Okay. So what we are doing today is mainly the lipid transport. All right, where we'll talk about lipoproteins, especially LDL, HDL, VLDL, the associated lipoproteins, and of course we have to discuss the disorders. We have to discuss the disorders associated with them. Is that fine? Okay. So first of all, I welcome you to the YouTube channel Let's Crack NEET PG. That's the Unacademy channel for our NEET PG division. And we also have a Telegram channel, so you guys can follow us on Telegram to get regular updates about the class. The link for that is there in the description for this particular video. My name is Chetanya Mittal, MBBS, AIMS New Delhi, and I hope you guys have downloaded the Unacademy learning application. You might know that we have launched a 2-month batch for Unacademy, right? And in 2 months, for all 19 subjects, especially for those students who are preparing for their PG entrance, we are giving a test and analysis, that is a TND batch, test and discussion and test and analysis, right? Besides that, we have a grand test series that is going to go on starting from 31st July, and we're having 12 grand tests as part of the series. And of course, we are having 3 hours of revision with the top faculty for all the 19 subjects, right? So we are going to give 3 hours to every subject, and the schedule is made till 7th of September before your NEET PG examination. Okay? We have a 2-month subscription so that you can just enroll in this batch, and if you use my referral code Chetanya10, you can get a 10% off on this particular subscription, and it will cost you around about 7,000. Similarly, if you go for the 1-year plan, you will get an advantage of 2 months. That is, 2 months are free. Again, if you use my referral code Chetanya10. Please use the right referral codes. Please do not go wrong with the referral codes. We have batches going on for next. We have batches going on for profs, right? That is your university examinations. And then we have batches for FMGE. Of course, we also have batches for INI, CET, NEET, PG, all of those examinations. Any examination related to medical entrances, we have a course for that. All right. Recently, we've launched a 4-year subscription, which means that those students who are right now in their first year, they can take this subscription, and then they don't have to think about anything till their PG entrance. And it's the most advantageous thing to take if you are right now in the first year. Okay, if you take it later in the second year, okay, you are losing 1 year of access, and you will almost have to pay that much only. Trust me, this is true for all platforms. Okay, so go for the 4-year plus or iconic subscription. In the iconic subscription, the benefit is you also get prep ladder. All right, so you are getting both the live experience and the learning recorded experience. ठीक है, and कुछ subjects तो ऐसे होते हैं जो लोग सिर्फ prep ladder से पढ़ते हैं, कुछ subjects ऐसे हैं जो लोग सिर्फ और सिर्फ Unacademy से पढ़ते हैं, right? Okay, so 4-year plus is 60,000, but if you apply my referral code Chetanya10 here, you will get a 10% off. So this will basically cost you 54,000. Similarly, you can see for iconic, the referral code is already applied. It was 75,000, but it costs you 67,500. Here's the referral. Okay, and this is all a limited time offer. Okay, so these are the batches for various profs, and we have over 34,000 MCQs for you guys. And these are all the benefits of Plus. We recently also introduced audio doubts. That is, you can unmute yourself in the lectures if you are a Plus user on the Unacademy platform. On the YouTube platform, we have introduced a program called as the Bugs Bounty program. Okay, so if you report any inappropriate content in the video and you are the first one to report it, you can claim a prize. So that's an incentive for you guys to watch all the videos carefully and report any inappropriate content. All right. Okay. So yeah, let's go ahead. Right. Let's come to our topic. You guys are ready? Are you guys ready? Should we begin? Give me some indication in the chat that you guys are ready for this class. You guys are excited. And initially, this class was supposed to happen yesterday, but due to some scheduling issues, I am taking it today. I know some of you might have waited for this class yesterday. I'm really sorry about that. All right. So in the previous class, I had given you a complete overview of lipid metabolism. I told you where to read it from. I told you what to read. All right. We had covered fatty acid metabolism, ketone body metabolism, and metabolic fuels. If you have not done all this, you can go to my previous video for this. Today, what we are going to be doing is the more important aspect of lipid transport. Okay. So I'll just show you what all we had covered about fatty acid synthesis, where it is occurring, what is the mechanism involved, right? What is the regulation? Give me 1 second. Right, so we have done this, and I talked to you not only about fatty acids but also about acetyl CoA, also about about ketone bodies. That is the acetone, acetyl, acetone, right? And once we had discussed the ketone bodies, the entire process, I had talked to you about the metabolic fuel use, and I had shown you this table, right? So I hope you guys remember this table. It's very, very important. In case you did not take a screenshot the last time, do take a screenshot now. Okay? And then finally, coming to this, we had finished off this topic. Now, let's start with lipid transport. So I told you we'll have a separate session for lipid transport. So today what we are doing is lipid transport. There's a small cut on my tongue. So you guys might be feeling that there's a slight lisp in my voice. That's because of that. I'm not able to speak a few words because of a small cut on the right side. So really sorry about that. I hope you guys are able to understand what I'm saying. I'm trying my best to, speak clearly. Some of you might be feeling कि थोड़ा सा words जो हैं, they're remaining in the mouth. Wherever you don't understand anything, please stop me. Is that fine by everyone? थोड़ा सा motivation support दे दो यार. Your educators are teaching you with cut tongues because learning should not stop, na? Okay, so let's begin. So you all know what happens. You have Dietary fat. Dietary fat. You all know a little bit about the digestion and absorption of fats. I am assuming you know a little bit about the digestion and absorption of fats from your previous knowledge. So I'll give you a quick overview. Fat, cholesterol, all of it comes in. Okay. It is emulsified by bile. It is broken down into small constituents like fatty acids and glycerol by various enzymes. And ultimately, what you get are organized micelles because of the presence of all these bile salts and everything. You get organized micelles. All right. Thank you, Dr. Sameer Tandon. Good to know. I just feel it's reassuring that you guys are saying that. So you get something where the hydrophobic fat components are inside, and you have the bile salts and everything on the outside. Okay, I just changed the colors. That should be wrong. Okay, so you have what formation? Micelle formation. Right? So basically, you have the formation of micelles. Now, why are micelles formed? Micelles are formed solely for the purpose of transport. Okay, micelles are formed solely for the purpose of transport inward. So when these fatty acids and glycerols they get transported inward in the enterocytes, what happens is, let's say this is an enterocyte. This cell is a enterocyte. So once these reach inside, the whole process of digestion is reversed. The whole process of digestion is reversed. The reason is that you need to form here lipoproteins, okay, called as chylomicrons. So you're basically going to send the fatty acids and glycerols. You're going to combine them with proteins because only when they are combined in that form, they can be sent to the lymphatic ducts here. Please remember that all fat is absorbed. I mean, most fat is absorbed into the lymphatic duct. So here I am drawing it like this. This is, let's say, a lipid packed with protein, and together we are going to call this chylomicron. So it's a type of lipoprotein because it has lipid and protein. So it's chylomicron. So it goes inside, and now you have chylomicron particles after your diet in the lymphatic system. That's the first step that you need to have. Okay. So what happens? Fat comes in, it is digested, it is uh, made soluble in the form of micelles. It enters the enterocytes. Inside the enterocytes, it is again repacked as lipids and combined with proteins to form a lipoprotein called as chylomicron. Okay, chylomicrons enter the lymphatic system. First step is chylomicrons, they are going to enter the lymphatic system. Is that fine? Okay. So now what happens here is that once those chylomicrons they've entered the lymphatic system, see, let's try to understand. Let's say this is a let's say this is the lymphatic system. You all know ultimately the final lymphatic is the thoracic duct for most of the body. The thoracic duct it drains into the venous system of the body. Sorry, yeah, right. So fatty acids they are coated with what to form chylomicrons? The answer is they are coated with proteins. They are coated with proteins. Not fatty acids. Basically, fatty acids are again combined with glycerol. You again combine the fatty acids with glycerol. You form lipids, and then you form triacylglycerols, and then you add it to the proteins to form chylomicrons. All right, Charu. Sorry for the slight interruption. Someone was at the door. Okay. ठीक है. So next step, let's go to the what happens after this. Right. So I was telling you that these chylomicrons, I'm just drawing them in the red, I'm not drawing the lipid inside. These chylomicrons, they are going to enter the, this is a vein, alright. So they are going to enter the venous circulation. Now once they have entered the venous circulation, what will they encounter? What will they encounter? So let's say here I'm making a chylomicron. So what is inside? You all know inside there is triacylglycerol or triglycerides. So it is written as TGs. Okay, TG is triglyceride. TG is triglyceride. Now there is something called as ApoB48. Okay, try to understand this. Now I will introduce you to a new concept. What are apolipoproteins? Apolipoproteins is equal to lipoproteins minus lipids. Lipoproteins minus lipid. So what does that mean? That means that if this is a lipoprotein, this is the protein part and this is the lipid part. If you remove this lipid part, what is left is called as the apolipoprotein. So there are a lot of apolipoproteins that are there. So basically all the proteins that are taking part in the formation of a lipoprotein are apolipoproteins. Okay. So you have apolipoprotein C maybe C1, C2, and now you have B, right? Apolipoprotein E. Okay. Now, so apolipoprotein B is something, so it is written as ApoB. ApoB. It is written as what? ApoB. ApoB means that it is apolipoprotein B. ApoB means it is apolipoprotein B. Now, ApoB, it has two forms. ApoB48 and ApoE100. Now, what is the meaning of these two different forms? What is the meaning of these two different forms? Something that happens here is called as mRNA editing. There is something called as post-transcriptional mRNA editing. Okay. So post-transcriptional mRNA editing. Okay, so I'll just explain it to you. Let's say there's a gene for apolipoprotein B. There's a DNA for apolipoprotein B. Let's say it gets transformed to form mRNA. Let's say this mRNA, it has a total of 300. Let's say, for example, I'm just telling it to you to give you an example. Let's say it has a total of 300 nucleotides or 100 codons. That's fine. Let's say it has 100 codons. 100 codons for amino acids. Okay. So what happens is this mRNA that is present, which has 100 codons, in the intestine, what happens to this mRNA is that this mRNA gets a something called as post transcriptional mRNA editing. which means that there is some editing that is done, because of which after 48 codons, there is a stop codon. A stop codon is added. Okay, and then this gets translated. This gets translated. So that means that only 48% of the RNA is being translated. That means that only 48% of the protein is being translated. So that means it's a smaller protein. So now you understand what is ApoB48 and what is ApoB100. Okay, so right now I'm telling you ApoB48, it is formed where? In the intestine. Because in the intestine there is absorption of fats. Because in the intestine there is absorption of fats, so it is going to be present in chylomicrons. It's going to be present in chylomicrons. So now let's redraw chylomicrons. Let's say this is a chylomicron. Inside, what do you have? Triglycerides. Outside, you're going to have which proteins? ApoB48. Is that clear to you? Is that clear to you? It should be absolutely clear in your mind. Okay? Is that clear to you? Any doubts? So you understand how ApoB48 comes here. How ApoB48 comes here to the chylomicrons. Is that clear to you all? Okay. So ApoB48 means that it is, here you can please note down that ApoB48, it is unique to chylomicrons. It is not present anywhere else. It is only present in chylomicrons. ApoB48, it is not present anywhere else. It is only present in chylomicrons. Okay, so you understand why it is called 48, because it is only 48% of the protein. It is only 48% of the protein. Okay, got it? Because only 48% of it is translated because of post-transcriptional editing due to a mutation. Not a mutation, but an editing, right? Cytosine gets converted to uracil. Cytosine gets converted to uracil, and because of that, there is a stop codon. It is very similar to a nonsense mutation, but ApoB48 is a functional product. ApoB48 is a functional product. It is not a non-functional product. Okay. So you might ask me, where is ApoB100 found? Where is ApoB100 found? When I am teaching you, I will tell you. So this is synthesized by the intestine. This is synthesized by the liver. Okay. And it is found in LDL and VLDL. It is found in LDL and VLDL. Got it? So this is a very important concept. A lot of times examiners like to go after this. Hmm? Okay. If you want to understand more about the synthesis, I'll tell you one more thing, because I know that you guys are very curious. तुम्हारा दिमाग चलता ही रहता है, तो मैं बहुत सिंप्लीफाई करके बता रहा हूँ. ये रहा स्मूथ एंडोप्लाज्मिक रेटिकुलम. Okay, so what we do is we put lipid inside it. We put lipid inside it, and there is protein inside it. Which protein? ApoB48. There is an enzyme here. There is an enzyme here called as MTP. Okay, not medical termination of pregnancy, but microsomal triglyceride transfer protein. Microsomal triglyceride transfer protein, and it is responsible for the synthesis of chylomicrons. Okay, because it will combine both these elements and it will lead to the formation of chylomicrons. So just understand this, and then everything becomes simple. Then these particles, they are going to enter the blood. They are going to enter the blood. So right now this is a nascent chylomicron. Okay. Now what happens here is, this chylomicron, why am I calling it nascent, is because it does not have all the apolipoproteins it needs. इसको जिंदगी में अपना काम करने के लिए कुछ और apolipoproteins चाहिए, right? And who is going to give it to them? Who is going to give it to them? That is another lipoprotein called as high density lipoprotein called as HDL. So here let me just draw something. So this is HDL, high density lipoprotein. It has two specific lipoproteins. Okay, I'm drawing triangles. Apo lipoprotein C2 and Apo E. So what it does is it transfers these apolipoproteins to the chylomicron. Okay, so ultimately what do you get? So you have ApoB48, right? You have ApoC2, and you have one more, that is ApoE. Now my job is to tell you what are the functions of these various apolipoproteins. Why is ApoE important? Because it is recognized by the liver. Recognized by the liver. Why is apolipoprotein important? Because liver receptors, they do not know what is a chylomicron. They only know apolipoprotein E. They do not know what is a chylomicron. They only know what is apolipoprotein E. Right? Now, why is ApoC2 important? ApoC2, it is important for the activation of lipoprotein lipase. I will tell you why they are important. Okay. Apolipoprotein 2 is activator of LPL, lipoprotein lipase. Two things. ApoC2 is an activator of lipoprotein lipase, and ApoE, why is it important? Because it is recognized by the liver. Who is going to give them these apolipoproteins? HDL. Who is going to give them these apolipoproteins? HDL. So now let's go back to the complete picture. What is happening? Fats were absorbed, chylomicrons were formed, they came to the blood. In the blood, from HDL, they got some additional apolipoproteins, and now they are going to go and reach the liver. Now they are going to go and reach the liver. Okay, so what happens here is, let's talk about two tissues here. So let's say this here, this is fat, and let's say this here is this is your chylomicron. This here is your chylomicron with Apo C2, Apo B48 and Apo E. Inside it has triglycerides. Now what the fat wants is, fat wants these lipids for its storage. The fat here, the adipose tissue wants these lipids from the blood. It has to go to the fat, it has to go to the adipose tissue. So it has an enzyme here called as lipoprotein lipase. It has an enzyme here called as lipoprotein lipase. Lipoprotein lipase. So what lipoprotein lipase is going to do is, lipoprotein lipase, it is going to extract all the fatty acids. It is going to extract all the fatty acids. It's going to try to remove the triglycerides. It's going to remove the triglycerides from the lipoprotein. Okay, so what is happening basically here is, these triglycerides, with the help of ApoC2, I told you ApoC2, it is an activator. It is an activator of lipoprotein lipase. So what is happening here? You can make out these triglycerides, they are going to get here and they're going to get stored as fatty acids in the adipose tissue. What you have left is called as the chylomicron remnant. What you have left is called as the chylomicron remnant. So you might come across this term sometimes, chylomicron remnant. After the lipoprotein has given its lipids to the adipose tissue, it is called as chylomicron remnant. It does not have ApoC2 because ApoC2 is used for activation of lipoprotein lipase. It has ApoE and it has ApoB48 and some amount of triglyceride, but it is less than a chylomicron. Okay, some amount of triglycerides. Got it? Got it? Fatty acids, they are stored by the adipose tissue. They are stored by the adipose tissue. And the glycerol, it may be used by the liver. Okay, got this point? Why is apolipoprotein C2 important? Now what happens is, so this ultimately, this apolipoprotein C2, it came from HDL and it is returned to HDL. It is returned to HDL. If you are wondering what is the fate of apolipoprotein C2, it is ultimately returned to HDL because it was borrowed from HDL. HDL से ही तो लिया था, तो अब HDL को जाके वापस कर देंगे. ठीक है. तो अब जो काइलोमाइक्रॉन रेमिनेंट है, because of this ApoE, there are receptors for apolipoprotein E. So liver is able to take this entire molecule up and it takes up all the cholesterol. Okay. Remember, this lipid core, it has both triglyceride and cholesterol. Triglyceride is taken up by the adipose tissue and the cholesterol and everything is taken up here by the liver with the help of apolipoprotein E. ठीक है. तो लिवर के पास क्या आ गया? Chylomicron remnant. So this is about the metabolism. This is about the metabolism of chylomicrons. Clear? So this is called as cholesterol transport. This is called as cholesterol transport. Is this much clear to you? Is this much clear to you guys? First tell me this. Is this much clear to you guys? Okay. So this is called as cholesterol transport. which means that you are transporting lipids, that is triglycerides, plus cholesterol from the intestine to the adipose tissue and liver. ठीक है? So whenever you are studying biochemistry, just try to understand what the body is trying to do, and then try to fit in the story inside it. Lipoprotein lipase, it is present in the adipose tissue. You're asking where is lipoprotein lipase present? It is present in the adipose tissue. Okay. Lipoprotein lipase, it is also present in the muscle. It is also present in the muscle sometimes, right? Sites of location of lipoprotein lipase, it is present in the lipids, that is the adipose tissue, sorry. It is present in the adipose tissue. It is present in the cardiac muscle. It is present in the skeletal muscle. So just understand what the body is trying to do. It is trying to absorb lipids from the intestine and deliver them to either the adipose tissue or the liver. Cholesterol mainly goes to the liver, all right? Or it will stay in the blood. ठीक है, because liver has to synthesize a lot of molecules using cholesterol. Remember, cholesterol, it is used for the biosynthesis of a lot of other molecules, so it has to go to a lot of places. Cholesterol is required by the body in good amount. ठीक है, so see, understand, intestine ने क्या किया था? एपोलाइपोप्रोटीन B48 प्रोवाइड किया था. लिवर के लिए हमने कौन सा एपोलाइपोप्रोटीन यूज किया? ApoE, because it has receptors for ApoE. And for adipose tissue we required ApoC2 because we had to activate lipoprotein lipase, which is required for the breakdown of chylomicrons. So once you know what is to be done, ये apolipoproteins याद रखना is a logical thing. It is a logical thing. उसमें कुछ सोचना नहीं है, ठीक है? Just know कि अच्छा क्या, पहले मन में सोचो क्या काम क्या करना है यार. अच्छा, इंटेस्टाइन में फैट आया है, उसको पहुंचाना है, ठीक है. उसके लिए क्या क्या चाहिए होगा? एक तो काइलोमाइक्रॉन को लाइपोप्रोटीन दे दो, एपो बी 48. उसके बाद एचडीएल से एपो C2 और एपो ई ले लो. क्यों ले लो? क्योंकि लिवर के लिए एपो ई चाहिए, काइलोमाइक्रॉन रेमनेंट डिलीवर करना है, एडिपोज टिश्यू को हमने अंदर का माल डिलीवर करना है. उसके लिए हमें एक्टिवेट करना पड़ेगा लाइपोप्रोटीन लाइपेज को, और लाइपोप्रोटीन लाइपेज के एक्टिवेशन के लिए क्या चाहिए? एपो C2. तो एपो C2 भी ले लो एचडीएल से, ठीक है. एपो C2 बाद में वापस लौटा दो एचडीएल को, एपो ई के थ्रू लिवर में लाइपोप्रोटीन चला जाएगा. ठीक है. नेक्स्ट, लेट्स टॉक अबाउट रिवर्स कोलेस्ट्रॉल ट्रांसपोर्ट. रिवर्स कोलेस्ट्रॉल ट्रांसपोर्ट माने लिवर को भी अपनी चीजें पूरी बॉडी तक पहुंचानी है. ओके, लिवर हैज नाउ टेकन अप कोलेस्ट्रॉल, लिवर हैज नाउ टेकन अप द काइलोमाइक्रॉन रेमनेंट, but liver cannot infinitely keep storing all of this. So liver has to deliver these to the body. For example, if the adrenal glands, they want to synthesize a hormone and they want cholesterol, they might say कि अच्छा भाई liver थोड़ा सा cholesterol दे दे, है ना? So from the liver, the cholesterol has to go, or it might want to be secreted in the bile. In some way we want to remove it from the liver, right? So that is called as reverse cholesterol transport, where liver is synthesizing the lipids. रिवर्स. व्हाई इज इट रिवर्स? बिकॉज यहाँ पे लिवर में जा नहीं रहा है, लिवर से बाहर आ रहा है. ओके, शुरू करें. यहां तक समझ आया? बाकी सब जो बताया मैंने, एवरीथिंग एल्स आई हैव टोल्ड यू, आई होप यू हैव अंडरस्टूड दैट. ओके, Now, this is the liver. This is the liver. What does the liver have? The liver has cholesterol. The liver has triglycerides. So what does the liver do? It has smooth endoplasmic reticulum. I told you there is an enzyme called as microsomal transfer protein, right? Microsomal triglyceride transfer protein. And liver, it does not have ApoB48. Liver has ApoB100. Liver has ApoB100. Complete product. These two are going to go, and MTP is going to release a molecule called as VLDL. And it is going to have ApoB100. Okay, is going to have ApoB100. So those of you who have watched the video from the start, अब तुम्हें बहुत प्यारा समझ आ रहा होगा. अब तुम्हारा लॉजिक भी चल रहा होगा. Now again it has to go to the peripheral tissues where it will activate lipoprotein lipase. Where it will activate lipoprotein lipase. So from HDL, what all does it have to take up? From HDL, it has to take up two things. One is ApoC2 and the other is ApoE. ये तो HDL से लेना पड़ेगा. Right? So VLDLs, they are secreted into the blood by the liver. They have ApoB100. They have ApoB100, right? They have ApoB100. Okay. Now what happens? Now what happens to these VLDL molecules? See, please try to understand. There are two components to a lipoprotein. Lipid and protein. This is the low density component. This is the high density component. If the lipid starts to decrease, what will happen to the density? The density will increase, right? The relative density, it will obviously increase because you have decreased the lipid. That means that the ratio of protein to lipid becomes more. So protein is more, so the density increases. Now what happens? The VLDL, it goes through the body. VLDL, it goes through the body. है ना? So in the body, at various sites, with the help of ApoC2 and lipoprotein lipase, it is going to give away its triglycerides, right? So the level of triglycerides becomes less. The level of triglycerides becomes less. That's correct. Level of triglycerides becomes less. है ना? As a result, the density increases. As a result, the density increases. है ना? तभी तो VLDL से very low density lipoprotein से low density बनेगा. है ना? या low density से intermediate density बनेगा. It means that the VLDL, it is depositing its triglycerides to the body, to the peripheral cells, and it is getting converted to LDL and IDL. LDL and IDL. Is that clear? ठीक है, एक और एक्सचेंज होता है. This is one more exchange that occurs that sometimes might be asked to you. What happens is, ये रहा VLDL, ये रहा HDL, VLDL, HDL, है ना. तो VLDL के पास ट्राइसाइल ग्लिसरॉल है और कोलेस्ट्रॉल है ना. इसके पास भी ट्राइसाइल ग्लिसरॉल है और, तो VLDL एंड HDL आपस में क्या करते हैं, दैट what they do is they transfer the triacyl glycerol from the VLDL to the HDL, and the cholesterol goes from HDL to VLDL. So HDL says तू मेरा कोलेस्ट्रॉल ले ले and मैं तेरा triacyl glycerol ले लूंगा. This exchange is done by cholesterol cholesterol ester transferase protein, CET. That's an exchange that takes place. ठीक है. Okay, so now what happens here is, so slowly you will understand that VLDL, it loses its triglycerides. Because of that, it was earlier very low density. The density increases but not as much as HDL. So it becomes a LDL, that is a low density lipoprotein. It becomes a low density lipoprotein. So now what happens is that low density lipoprotein, see, remember this still had ApoB100 and this has ApoB100. Okay. So now what LDL does is, in the body, there are certain places like peripheral tissues or liver. They have receptors for ApoB100. They have receptors for ApoB100. So they can take up this LDL. They can take up this LDL. Okay. Now these receptors, they can bind to ApoE also. Okay. Therefore, these receptors are called as, now you get the complete picture. They can either bind to ApoB100 or they can bind to ApoE. Okay, so this is called as ApoB100 / ApoE receptors. But they cannot bind to ApoB48. Please remember, they cannot bind to ApoB48. They cannot bind to ApoB48. Okay, this is called as receptor mediated endocytosis. These are called as receptor mediated endocytosis. Got it? Receptor mediated endocytosis. Any doubts so far? Any doubts so far? Okay. So I told you chylomicron, I've told you remnants of chylomicrons. I've told you VLDL. I've told you LDL. I've told you IDL. Now you must be wondering, where did HDL come from? High density lipoprotein कहाँ से आया? Okay, so what is the characteristic feature of high density lipoprotein? It has a special apolipoprotein called as ApoA1. ApoA1 has a special uh, apolipoprotein called as ApoA1. Okay, HDL particles, they are formed in the blood. How? Because see what happens here is liver और आपका intestine, they basically secrete ApoA1. They secrete ApoA1 into the blood. This is the blood. They secrete ApoA1. In the blood, whenever this ApoA1 finds some lipid particles, it is going to combine with them. It is going to combine with them to form HDL. Because it has very less amount of lipid, because it is taking up lipid from the blood, therefore it has very high density. It has very high density. It has very high density. That is how it is being formed. Okay. They're also formed within the liver and the intestine, but a lot of them are formed in the blood. Okay. And what is the function of HDL? It is mainly supplying. It is mainly supplying ApoC2 and ApoE to the chylomicrons and the VLDL. Alright. And because of their very important role everywhere in the reverse transport especially, they can help in the esterification of cholesterol. They can help in the esterification of cholesterol, and they can also take up unesterified cholesterol because they have an enzyme called as LCAT. ठीक है, छोड़ दो अभी वो सब चीजें. I'm not going into those details because then सब कुछ complicate हो जाता है. Okay. So reverse cholesterol transport तब complete होता है आपके mind में when you understand that how the cholesterol is moving. Remember I told you about this transfer. Now I'll give you the complete picture. मैंने जो तुम्हें थोड़ा पहले बताया था that was slightly incomplete. Reverse cholesterol transport में क्या हो रहा है basically? To be very precise, पेरिफेरल टिश्यू से कोलेस्ट्रॉल वापस जा रहा है. कैसे जा रहा है? दैट गोज वाया HDL. Remember, HDL used to exchange its cholesterol. HDL used to exchange its cholesterol with VLDL. So that is why VLDL I said was part of the reverse cholesterol transport. So HDL here, it is very crucial in reverse cholesterol transport. That is why it is the good lipoprotein. That is why we want more HDL. That is why we want more HDL. Because it helps in reverse cholesterol transport. Cholesterol goes from peripheral cells to HDL. From the HDL, it goes to the liver. From the liver, it is excreted in bile. Or it can be diverted for hormone synthesis. ठीक है? HDL. And estrogen, it raises HDL levels. Estrogen, it increases the levels of HDL. Estrogen increases the level of HDL. That is why females, they have a better cardiovascular profile. ठीक है, थोड़ा सा कॉम्प्लिकेट हो जाता है ये सब. And because I am giving you a slight overview rather than a complete detailed picture, कहीं कहीं पे you guys might feel that there are certain lacuna. Okay. So what I'll do is I'll make a few tables and I'll summarize some stuff for you. First of all, what are the enzymes in cholesterol transport? Okay, let's start. First, I talked about MTP. है ना? It's present in the smooth endoplasmic reticulum, microsomal triglyceride transfer. What is the function? Formation of chylomicrons or VLDL. How? It combines ApoB48 in the intestine or ApoB100 in the liver with proteins. Sorry, with lipids. Okay, that's the function. Second enzyme, lipoprotein lipase. Activated by ApoC2. This is also a question that can be asked. Activated by ApoC2. What is the function? Degrades triglycerides in circulating lipoproteins. Okay. Similarly, you also have a hormone sensitive lipase. You have a hormone sensitive lipase. What it does is it degrades the stored triglycerides in the adipose tissue. Okay. There's another enzyme, hepatic lipase. Okay. What it does is it degrades the triglycerides in IDL, intermediate density lipoproteins. Okay. Then I told you about one protein, CETP, cholesterol ester transfer protein. What it does is it helps in exchange of cholesterol esters. I'd given you an example above. Next, LCAT. What is LCAT? Lecithin cholesterol acyl transferase. Esterification of plasma cholesterol. Esterification of plasma. So those are some of the enzymes you should know about. Okay. So what I'll do now is, अच्छा मेरे को ये बताओ किस किस के मन में बिल्कुल सब हलवा हो गया है, किस किस के मन में बिल्कुल सब हलवा हो गया है. See, this is one topic that you have to read twice or thrice to understand it. Okay, I'll send you a few pages. Just read those at least. Okay. Acha. So to summarize some of the apolipoproteins, to summarize some of the new all the apolipoproteins, I'm going to tell you something. ओके वैसे बहुत सारे होते हैं, वी आर ओनली स्टार्टिंग द मेजर वंस. ठीक है, लेट्स स्टार्ट विद एपोलाइपोप्रोटीन A1. Okay, A1. So apolipoprotein A1, it is found only on HDL. HDL is also called as alpha lipoprotein. Please remember, it is also called as alpha lipoprotein. That will help you guys remember A alpha. A alpha. This much is clear? Right. It is found only on HDL, and one enzyme that it activates is LCAT. It activates LCAT. I just told you HDL, it is very important for transfer of cholesterol, है ना. Overall जो reverse cholesterol transport वगैरह है उसमें it is very important. So LCAT, it is responsible for esterification of cholesterol. So it is an activator of LCAT. जहां भी इसे cholesterol दिखेगा उसे एस्टेरिफाई कराने में हेल्प करेगा. Right, that is about A1. Then B48 and B100. Let's talk about it. Where is it synthesized? Intestine. Where is it synthesized? Liver. What it does is it binds the LDL receptor. Remember, there was a ApoB100 and ApoE receptor. Chylomicron secretion. It is important for chylomicron secretion into lymphatics. It is important for this secretion. So where will B48 be found? It is found in chylomicrons and remnants of chylomicrons. So I'm writing chylomicron and remnants. Where will ApoB100 be found as? It will be found in VLDL, it will be found in IDL, it will be found in LDL. Sorry, found in LDL. Ultimately, you have third one, Apo C2. Apo C2. ठीक है. So first of all, it is C, so it is a cofactor. Cofactor for cleavage. Why? Activates lipoprotein lipase. Activates lipoprotein lipase. So where all will it be found? Is it found on chylomicron? Yes. Is it found on chylomicron remnant? No, because chylomicron remnant has given up its C2 to HDL. Similarly, it is also not found in LDL. It is found on VLDL, and of course, HDL is a reservoir of ApoC. Last, ApoE. ApoE. Okay. Mediates uptake by everything except LDL. E for everything, E for except. So please remember, it is found everywhere except LDL. Got it? Any doubts so far? Guys, tell me one thing. अभी तक जो समझाया, अभी तक जो बताया, क्या वो समझ आया? Everything that I've told you so far, everything that I've explained so far, do you think that it was helpful for you so far? ना अभी तक जितना बताया. उसमें से did you feel that you were able to extract some knowledge? Yes? Abhi, I'll tell you some more things. Around, I'll take 15-20 more minutes and wrap up some of the other concepts here. Okay. What I'm planning is dyslipidemias we'll do as a separate session. For dyslipidemias, let's have a separate session. Abhi, class is not over. I have some more things to explain, but I just wanted to get your opinion. For dyslipidemias, we'll have a separate session. है ना? ठीक है. So right now what I'll do is yes, of course, very important is that you revise again. But before revising, I'll tell you what to do. After you have watched this lecture, what I will do is on my Telegram, I'll send some 5 to 7 pages. Okay, read those and then make your own notes out of it. I'll also send these notes that I'm making right now in the class, right? I'll send 5 to 7 pages. Just read those ones. Okay. So they are the exact information that you need for this entire topic. There will be some things extra that I have not taught you right now, and usme se you will be able to understand all of that also. Alright. So there is some topic like macrophage का जो scavenger receptor वाला concept होता है, oxidized LDL which leads to atherosclerosis. Those are some concepts that are also covered here. But right now I haven't touched on those. I've just told you basics. I've told you what you need from a mental picture to understand this topic. Okay? I'll do it very soon. In a day or two, I'll do it. Not an issue. Right now, what I'll do is I'll summarize the functions of all the lipoproteins for you. Functions of lipoproteins. Let me summarize for you. Okay. Okay. So first, try to understand that lipoproteins, what all are they made up of? What is the composition of lipoproteins? What is the composition of lipoproteins? So you tell me, what all do you think is present in a lipoprotein molecule? What all do you think is present in a lipoprotein molecule? Tell me. Okay. So I will go step by step. I will give you some data. First of all, they have triacylglycerol, right? Triacylglycerol or triglycerides. Secondly, they have phospholipids like lecithin. Third, they have cholesterol and cholesterol esters. And lastly, they have protein. Right? So that's the composition. Okay. Of course, it's lipids proteins, but I just wanted to tell you that there are 3 types of lipids: triacylglycerols, phospholipids, and cholesterols. Okay. And in every sort of lipoprotein, this ratio is variable. So do you guys want me to tell you कि क्या composition रहता है? Okay. So let's just quickly cover the composition of individual lipoproteins because that can also be asked as a question. Okay. So first let's start with HDL. HDL is high density. Now why is it high density? You guys will tell me. Because it has more. Which of the components will be more in HDL? Which of the components is going to be the maximum in HDL? Which of the components is going to be the maximum in HDL? Proteins, right? So maximum is proteins, right? Forget about phospholipids. They're not very important. Followed by cholesterol fraction. Okay. Cholesterol and cholesterol esters. Followed by lowest is triacylglycerol. So this you know why. You understand this also? Remember I had told you in one of the slides above that there's an exchange between VLDL and HDL, where HDL takes up the cholesterol and VLDL takes up the triacylglycerol. So cholesterol is more here and triacylglycerol will be more. And you all know that in the reverse cholesterol transport and everything, for that the role of HDL is much more. So what is the order for HDL? Proteins, almost 50%. Almost 50% is proteins, right? A good fraction is proteins. 25% is cholesterol. Almost 25% is cholesterol, and only about 5% is triacylglycerol. Rest is of course your phospholipids. Okay, so easy to remember. First write 50, then half it, then divide it by. ठीक है, मतलब जैसे भी याद रखना रख लो, और ठीक है छोड़ो. Next let's talk about LDL. Okay, let's talk about LDL. Now before we talk about LDL, let's talk about VLDL. Let's talk about VLDL. What do you think is the highest here? What do you think is the highest here? Of course, triacylglycerol. That's why it's very low density, because there is so much triacylglycerol that you cannot even imagine. So much. Okay, and it has very less protein. It has very less protein, only 2 or 3%. Sorry, no, I'll tell you later. VLDL, it does not have 2 or 3%. It has slightly more, 5%. Then after that, of course, it also has a good level of cholesterol. So at the second position, you have cholesterol. Phospholipids I'm not touching. Phospholipids I'm not touching. Then it is 2. No need to remember the percentages. So yeah, what I was telling you, chylomicrons. Chylomicrons, they have come from the intestine. They are pure fat. They are pure fat. ठीक है, उनमें बहुत ज्यादा ट्राइसाइक्लिसरॉल है. Over 90% of it is triacylglycerol. And very less amount of cholesterol, and very less amount of phospholipid or protein or all of this. Almost very less. Okay. What is left is LDL, right? So LDL is having almost 50% is cholesterol, right? Almost 50% is cholesterol, followed by, so phospholipids and proteins are almost equal, but the triacylglycerol content is relatively less. Yeah. So by chance, if this is asked anywhere, you should know the trends. You should know the trends. No need to remember the percentages. Okay. One more thing that is sometimes asked is the electrophoresis picture. Okay. So how do you think this would work? If there are more protein, there is more. So what is the charge on proteins? Generally, what is the charge on proteins? What charge are you assigning to proteins? Generally, it has negative charge. It can also have positive charge, but generally it has negative charge. So more the protein, more is the migration. More the protein, more is the migration towards positive terminal. That is anode. Towards positive terminal, that means anode. So let me draw a gel electrophoresis plate. Okay, let's say this is the gel electrophoresis plate. Here you have the cathode, that is negative. Here you have the anode, that is positive. Correct? So what is the mobility going to be like? HDL has the maximum protein. HDL has the maximum protein. So the band for HDL is going to be somewhere here. This is the band for HDL. Okay. Lowest mobility, maximum fat is which? Maximum fat, सबसे ज़्यादा fat, सबसे कम protein किसमें है? Chylomicron. So it's almost going to be there at the originate site. It almost has zero mobility. Okay. Then you have which protein? LDL and VLDL. Okay. So what should have happened is you should have VLDL and then LDL. That is what you were expecting, but that is wrong. That is wrong. Okay. Just just think about it for 1 second. HDL had how much percentage of protein? I told you. HDL almost had HDL almost had 50% of protein. Between LDL and VLDL, who has more protein? Who has more protein? LDL or VLDL? LDL or VLDL? Who has more protein? LDL has more protein. I told you VLDL, it has relatively very less protein. Okay. So VLDL should be first and then you should have LDL. But that is not the case. The order is reversed. Okay. So the order is LDL and VLDL. That is that is because of the separation technique that we are using. That is because of the separation technique that we are using. No need to get into the details, but if the electrophoretic, if this is a simple uh, electrophoresis or an electrophoretic mobility chart, this is supposed to be LDL and this will be VLDL. So please remember, VLDL is coming between LDL and HDL. That is why HDL is called as, oh, this is called as alpha lipoprotein. This is called as alpha lipoprotein because it goes the farthest. It is an alpha. This is beta, and VLDL is called as pre-beta. It is called as pre-beta because it is before beta. It is finishing before beta. And this here, of course, is the. So this electrophoretic mobility chart can also be asked to you. To be able to answer, accordingly, you will have to answer. Is it clear with everyone? Just keep this in your mind. Spend 2 minutes and absorb this. Meanwhile, let me absorb water. Okay, done. All clear. Can you guys give me a thumbs up? Any doubts? Okay, so Charu has given a thumbs up. The rest of you, do you guys understand? Okay, let's go. Functions of lipoproteins. है ना? Let's talk about it first. Chylomicron. The function of chylomicron, I told to you guys very clearly. When I said chylomicron, what happens? Goes to the adipose tissue and delivers what? Triacylglycerols, right? After that, you have the formation of chylomicron remnants. They will go to the liver, and what do they deliver to the liver? They will give to the liver cholesterol. That is what they are mainly delivering to the liver, right? So that is the function of chylomicron. They are delivering glycerides to tissues, and cholesterol goes to the liver in the form of chylomicron remnants, right? Second, VLDL. What happens? Secreted by the liver. So it delivers endogenous or hepatic triglycerides to the tissues. Nothing complex. Those that are synthesized by the liver. Okay? Alright. VLDL VLDL, it undergoes degradation to form IDL, and IDL returns triacylglycerols and even cholesterol to the liver. That's also the role of IDL. Okay. Now, last two important ones, HDL and LDL. So first, let's do LDL. How is LDL formed? By the by the delivery of triglycerides to the peripheral tissues. VLDL gradually converts to LDL. VLDL gradually converts to LDL, right? Because of the role of lipases. Because of the role of lipases. Okay. So what happens? Hepatic cholesterol, the cholesterol released by the liver, it undergoes receptor mediated endocytosis. Receptor mediated endocytosis, right? And there is a special form of LDL called as oxidized LDL, right? So macrophages, they have something called as scavenger receptors. Macrophages, they have scavenger receptors. So this component is implicated in atherosclerosis. This component of LDL is implicated in atherosclerosis, especially oxidized LDL. So LDL is bad cholesterol. LDL is bad cholesterol. HDL, reverse Please remember, which is associated with reverse cholesterol transport. Your answer should be HDL. And because it transports in a reverse manner, it is good cholesterol, right? What it does is it transfers from the peripheral tissues to the liver. Cholesterol, right? Other functions, it also helps in supplies ApoC2 and ApoE to other lipoproteins, like I already told you. Okay, and you might have heard some people say alcohol is good, है ना? You might have heard some people say that alcohol is good for health. Why do they say that? Because alcohol increases HDL. Okay, but only when you have it in controlled amounts. Only when you have it in controlled amounts. Okay, everything you have to look at it from a holistic perspective. You can't simply say just because acha alcohol increases HDL, it is healthy. Now, you have to look at all the effects of something and then you can make a conclusion about something. You can't simply say that अच्छा alcohol HDL बढ़ाता है, ठीक है, very good, बहुत अच्छा है. That's not the only way, right? But this is one place where alcohol is good to some extent, and of course HDL is good lipid. So guys, ऊपर थोड़ा concept mess up हो गया था when I told you reverse cholesterol transport. मैंने तुम्हें थोड़ा सा गलत बता दिया था. So what happens is just make that correction in your minds. Intestine, see, let's say this is peripheral tissue. What is this? This includes everything. This includes muscle. Let's say this is fat. So I'm drawing muscle, right? So this is basically peripheral tissue. Now peripheral tissue, it has two ways to obtain triglycerides. It has two ways to obtain triglycerides or lipids, right? What are those two ways? Dietary fat goes to intestine, and this is basically coming to the peripheral tissue. That is called as exogenous fat. This is exogenous fat coming via ApoB48 wala pathway, and through the chylomicrons. Another way is endogenous fat. Okay, endogenous fat. This is coming via the liver, right? In you through VLDL and LDL. Through VLDL and LDL. Now, there's a mediator called as there's a very good mediator. I'm drawing it in green. That is going to take cholesterol from here, from the peripheral tissues, from the blood, and transport it back to the liver. So basically, this part from where either it is exogenous or endogenous fat does not matter. Exogenous or endogenous fat, it is coming to the peripheral tissues, that is bad. That is cholesterol transport. But when it is going back from the peripheral tissues to the liver, and then the liver has the option to secrete it in bile, it can go out of the body. That is called as reverse cholesterol transport. Understood? Got my point, all of you? Is all clear? Everything's clear? Any doubts? Okay, so I summarized everything for you here. This is the overview of the entire lipid transport. After this, there is a topic of dyslipidemias. Okay, first of all, any doubts. Any doubts that you have, please express them right now. Any doubts that you guys have, please tell them to me right now. Otherwise, please write down no doubts. Right, no doubts. Okay. So next lecture, I'll take a small lecture for this on dyslipidemias. There I will tell you about A beta lipoproteinemia. There I will tell you about Tangier's disease. And there I will tell you about the various familial dyslipidemias. There is a proper classification for this. What is that classification called as? What is that classification called as? Hyperlipidemias. Anyone has any idea what is that classification called as? So familial hypercholesterolemia. Okay, so I'll tell you about it there itself. It's called as Fredrickson classification. Okay, so we will discuss that. Fredrickson's. Till then, thank you so much guys for watching. Please remember to hit the like button for this video. Subscribe to the YouTube channel. Let's crack NEET PG.